topic I was asked to discuss with you today is the effect of stress on the endometrium and ultimately on implantation. So these are my disclosures, and the outline is again a few words about stress, CRH, implantation, and then some clinical data, not many, but the ones that we have. So we all know that stress, and especially this group of experts, knows that different types of data suggest that environmental events acting on the developing embryo in utero are crucial determinants for disorders later in life, a phenomenon that has been known as fetal programming. And indeed, prenatal maternal stress by different conditions may be associated with preterm birth. We'll hear more about that later today. Low birth weight, lifelong dysregulation, and metabolic and neurodevelopmental abnormalities, insulin resistance, type 2 diabetes, cardiovascular disease, and behavioral problems. So indeed, as part of this process, we can see that the effect of stress may differ according to the stage of development. Now, again, we know all that the stress system is constituted by the hypothalamic pituitary adrenal axis and is coordinated, is activated in a coordinated fashion as a response to stress, which influences the central and peripheral functions. Now, again, the main central regulators of the HPA axis is CRH and AVP, and we heard more about the production of these hormones. Now, if we look schematically, and again, so the hypothalamus produces CRH, which acts on the anterior pituitary to produce ACTH, which in turn acts on adrenal cortex to produce cortisol, which is converted to cortisol, and then there are, of course, feedback mechanisms. Now, stress through glucocorticoids and acts on different systems of the body, but today we will focus mostly on reproduction and particularly implantation because parts of it have been already addressed and will be addressed subsequently. So we know today, and Sarah gave some indications, that stress has a suppressive role on both the female but also on the male reproductive system. And so we heard more about that. I would like to skip to save time. So CRH is a 41 amino acid neuropeptide. It's synthesized in the hypothalamus, and it has receptors. And again, all this is work that came from Willie's group. So CRH-R1 in mammals, CRH-R2 in mammals, and CRH-R3 in catfish. So, So we know now that we have that CRH is produced in different parts of the body and in the organs that we are mostly interested today in my presentation, that is ovarian CRH, endometrial CRH, and placental CRH. So concerning the effect on pregnancy under physiological conditions, CRH promotes decidualization, assists blastocyst implantation and early maternal tolerance, and also blastocyst adhesion, implantation and invasion and maternal tolerance, and regulation of fetoplacental circulation. However, under pathophysiological conditions, may lead to failed or abnormal implantation and also to disorders like preeclampsia, IUGR, and preterm labor. And this exactly shows the same. So ovarian CRH, CRH is not detectable in oocytes from primordial follicles, but is abundantly expressed, CRH and CRH-R1, in mature follicles. And so its localization is in theca cells surrounding the ovarian follicles, in luteinized cells of the stroma, and in the cytoplasm of the oocyte. Placental CRH, large amounts, mostly during the latter half of pregnancy. Synthesis of CRH increases with advancing gestation and enters the fetus. Now, placental CRH is localized in maternal, high concentration maternal blood, fetal blood, and amniotic fluid. And CRH apparently participates in the maintenance of vascular tone, and it also is implicated in the modification of the high resistance non-pregnant circulation to a low resistance system in order to facilitate nutrient exchange through changes that happen at the endothelium of the uterine arteries. And this is again from a study from Dr. Makrigiannakis' group, showing that in the extravillous trophoblast, the concentration of CRH is significantly higher in cases of preeclampsia. So when this change in circulation does not occur or is impaired, then this may lead to a high pressure system which can be associated with preeclampsia. Now, intrauterine CRH is produced by the epithelial cells. The localization is in the stromal cells when differentiated to decidual cells, and CRH1 type 1 receptors are present in both epithelial and stromal cells. So this is a complicated scheme, but it clearly shows that the endometrial stroma produces CRH, produces prostaglandins, especially PGE2, and interleukins, and then they act via CRH or via progesterone, inducing decidualization or promoting decidualization. Now, implantation, we all know it's a process that goes on in the, starts in the fallopian tubes with fertilization and early cleavage, and then ends up after 5 to 6 days in utero where the blastocyst implants, and it's a very complicated process for which we know still not very much. And we have the phases of apposition, adhesion, and invasion. And this is, of course, a very critical moment, the invasion of the uterine epithelium by the blastocyst. Now, the process takes place in a given amount of days, which has been called historically the window of implantation, and one of the first people to discuss that was the late researcher from Greece working in Paris, Psychogios, and his colleagues. The efficiency of the process in the human species is remarkably low, and it is successful in no more than 30% of the cases, usually less, and the majority of pregnancy losses occur either prior to or during implantation. So successful implantation depends on basically two factors. Number one, the appropriate embryo development, and number two, the invasion of the embryo excuse me, into the decidualized endometrium. And of course, the maternal endometrial response to the invading fetal semi-allograft. And the characteristics is that of an acute aseptic inflammatory response, which is suppressed once the embryo is implanted. And it has been shown in rat endometrium that CRH on the implantation sites is 3.5-fold higher than in the inter-implantation region, indicating that CRH may participate or seems to participate in implantation. Now, in the fetal-maternal interface, CRH acts both on the embryonic trophoblast and the maternal decidual cells, and it induces the expression of Fas ligand, and this induces apoptosis in the activated cells, macrophages and others that express Fas, and therefore this protects pregnancy from these cells, from this reaction. So I will not spend time on Fas and Fas ligand. And here we can see again from this study the increase of CRH of the Fas ligand protein, which is aiming to induce apoptosis. Now, the trophoblastic CRH is the first tissue to differentiate. CRH controls proper trophoblast invasion by regulating the carcinoembryo CEA CAM1 expression, and therefore it seems to be to participate in the mechanism to limit the invasion of the endometrium. And as we can see here, again, the CRH is limiting the invasion and therefore participates in the regulation of the invasion. So trophoblast and endometrial CRH play a crucial role both in implantation and in the anti-rejection process that protects the fetus from the maternal immune system. And its critical steps is the, on one hand, promoting implantation and maintenance of pregnancy, and on the other hand is to control the invasion and the survival of the embryo. The endometrium restricts trophoblast invasion, and the extravillous trophoblast invasion leads to replacement of the endothelium, as I already expressed, in the uterine arteries so that we can have a low pressure system. Now, I'll show you some data indicating the involvement of CRH in spontaneous miscarriages, and as we can see in this study here, there are increased levels of intrauterine CRH and urocorticotropin-1, leading to abortion through the CRH-R1 induction of the complex Fas-Fas ligand. So if this is activated, then we have the reverse process. That means apoptosis in the trophoblastic area instead of having apoptosis in the monophages and other cells. And this was shown in this study, as we can see here, the normal, and in the abortion, the expression. So abortive deciduas contain leukocytes that are positive for Fas ligand, and extravillous trophoblast shows increased expression of Fas and increased rate of apoptosis. So as I said, the apoptosis is reversed and is observed now in the decidua. CRH and urocorticotropin were elevated in placental material obtained from the abortions. Another study that was done, it was using antalarmin, a CRH receptor type 1 antagonist, which exactly blocks this, the induction of apoptosis to the activated lymphocytes and the macrophages, and therefore leads, as it was shown in rats, that it does not lead to sterility, but it leads to decreased implantation and defective implantation. So antalarmin decreased Fas ligand expression and inhibited apoptosis of activated T lymphocytes. Female rats treated with antalarmin showed diminished Fas ligand expression and a 70% decrease in implantation sites. Nevertheless, antalarmin administration did not completely abolish implantation, suggesting that the process of other mechanisms is also operating. And this is in accordance with the fact that CRH and CRH-R1 knockout mice are hypofertile but not entirely sterile. Now, another interesting group of patients is women with repeated implantation failures. And this group has become particularly interesting in the context of in vitro fertilization. So my colleague and friend, Dr. Makrigiannakis, examined the question that, yes, large amounts of CRH lead to abortion, but is it possible that high amounts of CRH may help in securing implantation? So they performed a preliminary proof of concept study which included women with repeated implantation failures, 3 or more. That's the standard IVF procedure that they did. but then they took peripheral blood mononuclear cells which were isolated by centrifugation, and then these were treated either with, they were incubated with RPM1 supplemented with 10% SPS in the presence of human chorionic gonadotrophin and CRH for 48 hours, whereas in another group, they were only incubated with RPMI. And then this medium was, this suspension was, this cell suspension was administered in utero, in the uterine cavity, using an embryo transfer catheter 2 days after oocyte retrieval and approximately 3 days before blastocyst transfer. And these are the preliminary results that you can see here. And as you can see, the clinical pregnancy rate was appeared to be significantly higher, and the implantation rate in women that have been treated in intrauterine administration of these cells. And this is also shown here, that the clinical pregnancy rate and the implantation rate was significantly higher. Obviously, this is very preliminary and needs further confirmation. So it appears that the intrauterine administration of cells pre-treated with CRH and HCG may promote implantation. Now, what can be the possible explanations? CRH significantly increases IL-6 release from PBMC, significantly decreased IFN gamma levels in a dose-dependent manner. Immune and neuroendocrine systems interact at various levels, and in particular, either cytokines activate the hypothalamus pituitary adrenal axis, or CRH induces the release of beta endorphins. So increasing the evidence of a correlation between the immune and the endocrine system, that's something that is quite well known. So CRH added to the primary culture may increase IL-6 and decrease IFN gamma in a dose-dependent manner. Now, the question is, of course, whether we are ready to incorporate that into our clinical practice, and my answer would be no, but I think it's an interesting concept that needs to be further explored. So I would like to enrich my conclusions, dear colleagues, saying that CRH participates in the nidation of the fertilized egg by inhibiting local maternal immune response to the implanted embryo. CRH has a fundamental role in the mechanisms responsible for embryo implantation and maintenance of human pregnancy. Stress in utero, as this is depicted by CRH-involved pathophysiology, may affect implantation and early development. Abnormalities of decidual, trophoblast, and placental CRH have been implicated in several common diseases, in several common pathological conditions of pregnancy, including spontaneous abortions, preeclampsia, and intrauterine growth retardation. And increased ovarian CRH might be related to POF. So again, there are many hypotheses that need to be explored, but what I find interesting in this area, in this field, is that many, many years ago, there was always a discussion whether stress is associated with infertility or early pregnancy, early premature birth, and so on and so forth. And you know, we were saying, oh, we have no data. Now, some data seem to emerge, and I think stimulate us to do the proper research together with some of the basic scientists that are in the room in order to be able to provide reliable answers to this important question that needs to be answered. So finally, I would like to thank my colleagues, and particularly Professor Makrigiannakis and Dr. Kalpatsanidis, who particularly helped me in this presentation, my other colleagues in the University of Thessaloniki, and all of you for your attention. Thank you very much.